Hello, everyone. Welcome to our inaugural talk for UBCO's Diversity in Biology Speaker Series. We are very excited to have everyone attend, wherever you're calling in from. My name is Nathan Peltier. I'm an associate professor in biology and management, and I will be your host today. So this series is the result of a lot of hard work, and it would not be possible without our faculty coordinators, Dr. Karen Hodges and Dr. Robin Young, as well as our graduate coordinators, Shirley Yang, who is doing her master's in Dr. Clegris's Cellular and Molecular Pharmacology Lab and the Gibbon Lab, and Kat Favell, who is doing her PhD in Dr. Ford's Wildlife Restoration and Ecology Lab. Shirley, Dr. Hodges, Dr. Young, and I are calling in from UBC's Kelowna campus on the territory of the Silks Okanagan peoples, and Kat is joining from Edmonton in Treaty 6 territory. We acknowledge the many First Nations, Métis, and Inuit who have lived in and cared for these lands for generations and continue to do so today. And we invite you to acknowledge the Indigenous peoples who care for the lands that you are on today as well. With us, we are very pleased to welcome Dr. Yaxi who is currently working as a postdoctoral fellow at Health Canada, and who will join the Department of Chemistry at Carleton University as an assistant professor this July. Dr. Hu received her PhD from the University of British Columbia, Vancouver in 2018, majoring in food science. Her research is focused on the development of innovative analytical methods using spectroscopic, metabolomic, and genomic tools, as well as a microfluidic lab-on-a-chip platform for the detection of food safety, quality, and authenticity issues. She has published 26 peer-reviewed articles in top journals and won several prestigious awards, including the MyTax Outstanding Innovation PhD Award in 2016, as well as first place in the ACS Withicombe Charalambous Graduate Student Symposium in 2018. I'll hand it off to Dr. Hu in just a second, but first a quick housekeeping note. The audience is free to post questions in the Q&A throughout the talk. The questions won't be visible publicly until Dr. Hu has finished her presentation, but please do feel free to post them as they come to you. Because of the nature of this seminar series, you are free to ask Dr. Hu any respectful questions about her research or her career path so far. The seminar is being presented through Zoom's webinar mode, so you will not be able to turn on your cameras. So without further ado, please take it away, Dr. Hu.
Thanks, Dr. Pellier, for the kind introduction. Good morning, everyone, or I guess good afternoon for the people from the eastern area of Canada. It's my great honor to be invited to give a talk for this wonderful diversity in biology seminar at UBC Okanagan campus. So my talk today will be focused on application of omics and related tools for the analysis of food integrity. I'd like to firstly give a brief introduction about myself. So I originally came from the city where both the Summer and Winter Olympic Games have been hosted. Yes, you can guess it, it's Beijing. So this is a very beautiful city that has a lot of historical places of modern China. More about my training background, educational background. So I did my bachelor's in biological science from China Agricultural University. And then I moved to Vancouver, joined the University of British Columbia to finish my master's and PhD programs in food science. And then I did a postdoc training in the Department of Chemistry at UBC Vancouver before I joined Health Canada as a postdoc fellow. Now I'm currently working at Health Canada in their food research division located in Ottawa. And later on in July, I will be joining Carleton University, the Department of Chemistry, as an assistant professor. They have a specific food science program, so I'll still be working on food-related research. Majority of my training background have been focusing on food science, but I assume some of you are not majoring in food science. So before moving into my topic today, I'd like to briefly explain what is food science and what do food scientists do. So this is a really multidisciplinary field, ranging from chemistry to biology, from marketing to nutrition, from processing to sensory, everything you can think about for a food to be processed or produced from the farm to the fork can be covered by food science. And among all these different areas of research, the one that I'm particularly interested in are the food safety and quality related research. So for food safety and quality related areas, these areas can be covered by an umbrella term called food integrity. So this umbrella term basically covers 4 types of food related issues. They are the food quality issue, authenticity issue, safety issue, and defense issue. So these 4 types of issue have some intrinsic difference depending on if they occur naturally or there are some human practice involved, and also they have different outcomes. So for food quality and food authenticity issue, usually they don't cause any illness. And specifically for food authenticity, it's the motivation behind it is for extra economic benefits. For the food safety and food defense, both of these type of issues can cause some sickness. And for food quality and safety, these 2 issues occur naturally. No human intentional practice involved. While for food authenticity and food defense issue, there are people did that intentionally for them to be categorized into these 2 groups. In my previous research, I've been mainly focusing on solving the problems related to food quality, safety, and authenticity issues. So by only looking at the definition, it might still seem a little bit abstract. So I'd like to give you some more examples about the food integrity issues that we can see in our daily life. This case happened I think about 2 weeks ago. So FDA found Chronobacter and Salmonella in some powdered infant formula. This issue has caused huge recall globally for the infant formula powder, including in Canada. This is a food safety issue because Chronobacter and Salmonella can cause human sickness, but nobody add them into the food products intentionally. This is another food safety issue. So weed killer being found in cereal, breakfast cereal. So specifically the weed killer in this case was glyphosate. Glyphosate was used as a weed killer during the agricultural practice, but nobody wanted to present in the final food products. So this is also a food safety issue that occurred naturally. China dairy products contaminated by melamine was thought to be, this case happened in 2008 that caused thousands of injuries and death in a baby. So this case was thought to be a food safety issue because it caused a lot of hospitalization and death. But after digging into all the practice behind it, people found that it's actually a food fraud or authenticity issue. So melamine, it's a molecule that can boost the apparent protein concentration of dairy products to make low quality products look higher quality. So people added melamine to make extra economic benefits, but they didn't realize the melamine can cause renal failure in infants until the case happened. This is a typical food fraud case. So saffron, one of the most expensive spices in the world, has commonly been found to be contaminated by things like plant fibers or some colorants. This is like all of the materials are added into saffron to reduce the production cost. Spoilage problem, which can also cause some recall of the products. So usually the microorganisms cause spoilage, they don't cause human sickness. And those microorganisms occur or get into the food naturally. So this is categorized as a food quality issue. So seems like food integrity issues are very prevalent and difficult to prevent. There are a lot of strategies being proposed or being implemented to safeguard the food integrity in the supply chain. Here are the 4 major strategies that are promoted by the US FDA to tackle food safety issues, but they can also be generalized to solve food integrity issues. So including take the full advantage of advanced digital traceability instead of using the paper-based traceability to improve the efficiency and effectiveness in information sharing. And also by developing more innovative and smart analytical tools we can better detect the defective food products and get rid of them from the supply chain. There are a lot of new business models emerging recently, especially during the pandemic, a lot of online shopping being made available. So legislation really needs to keep updating to meet the new requirements of the new business models. Last but not the least, by promoting a food safety culture, or in general, the food integrity culture, defective food can be prevented from the beginning of their production. So I was trained mainly as a chemist. So my approach to help solve food integrity issue mainly focused on developing smart analytical tools. The overall research goal is to develop reliable detection methods that can facilitate the achievement of overall food integrity from farm to fork. So basically, I'm taking a detection-based approach. There are actually a lot of analytical methods that are available for detecting the defective food products. However, majority of the methods have some inherent disadvantages, including most of them are time-consuming, labor-intensive, the testing are costly. Sometimes the sensitivity or quantitative accuracy are not ideal. A lot of methods are very complicated to run, so they really need highly trained personnel to do the experiments. Also, a lot of methods are relying on large size laboratory-specific instruments that are not very ideal to be carried out when you're in the field or in some resources limited areas. So the specific objective for my research has been focusing on developing rapid, cost-effective, sensitive, accurate, portable, and or user-friendly methods to detect food integrity issues. To achieve my objective, I'm particularly attracted by this group of tools called omics tools. So here are the 4 typical omics technologies that developed based on the central dogma for initially for biological or for medical studies to analyze the DNA, the RNA, protein, and small molecules in complex sample matrices. In food integrity issues, so if you can recall the examples I mentioned earlier, the difference between defective food products and the genuine food products also lies within these 4 categories of chemical. So making these omics tools have high potential to be applied to solve food integrity issues. Actually, omics tools have much broader application in food science, such as there are researchers using omics tools to guide the production procedure, and also there are researchers using genomic tools to analyze the microbiome of fermented food. Food scientists even made a new discipline to acknowledge the studies that's been using omics tools. They call it the food omics. In my previous studies, I've been mainly using tools that belongs to or related to metabolomics and genomics. Here are the brief overview of the tools that I have been using in my previous studies. So they're grouped into two categories. One is based on small molecules or the chemical composition. The other is based on the DNA. I'll be talking about more details about each type of technology in my following slides, but here I just want to give you a brief overview. So the techniques for analyzing the small molecules include infrared spectroscopy, Raman spectroscopy, mass spectrometry. There's a typo, it should be spectrometry. NMR spectroscopy. For the DNA analysis, I've been using PCR and also some isothermal amplification, including RPA, the recombinase polymerase amplification, and LAMP, loop-mediated isothermal amplification. So let's take a look at the tools based on the chemical composition first. So the first group of tools I want to introduce are the infrared and Raman spectroscopy. These are regarded as a spectral fingerprint. So it collects the vibrational signals of functional groups in the chemicals. So here are the 6 major types of vibrational modes of functional groups. So because different foods have a unique combination of chemical compositions, and each chemical have a unique combination of the vibrational modes. So combine everything together, those signals can be treated as a fingerprint for the specific sample. It's just like the fingerprint is unique to each body, each human, each person. So the beauty of this type of technology is that they can be applied in a none or less destructive manner in a very rapid way. So usually the spectral collection takes a few minutes or even a few seconds, and very user-friendly. You simply shoot the laser onto your sample, collect the spectra, and the analysis is done. Another beauty for IR and Raman spectroscopy is that there are a lot of portable handheld-sized instruments available, making it very ideal to be carried out in resources-limited areas. This is one of the examples how I apply infrared spectroscopy for the authentication of ground beef meat. So we think that ground beef meat, once it's being grounded, the morphological features of beef meat is no longer there. So there are chances for frosters to mix some other things into the ground beef meat. And offal was the materials that we choose because offal, which are the internal organs of animals, so those are not consumed in a high demand in North America. So we think there are chances for the frosters to mix into ground beef meat. So for this project, we purchased the sample, we purchased the meat, the offal from the grocery store and prepared the ground samples in the lab. Then collected the FTIR spectra and using some chemometric models or machine learning models to analyze the results. So to try to differentiate, to classify the different samples and also to quantify the different, the level of the adulterants. After comparing the performance of different mathematical models, we eventually found the best models and developed this workflow for the analysis of new samples. So once you have a new ground beef samples that you suspect it might be adulterated, you can simply collect an FDR spectra of it. And we have developed a linear discriminant analysis model to tell if the sample is authentic beef meat or if it's adulterated with 100% accuracy. And if the sample is authentic, then the analysis is done. But if it's adulterated, and if you want to know more about what is being added, we have a 96% accuracy to tell you if it's adulterated with beef offal or with pork offal. And later on, if you want to know the concentration of the adulterants, and we have developed some, built up some regression models to do the quantification. So if the sample is adulterated with beef offal, we have a partial least squares regression model to quantify the concentration of beef offal with very high accuracy. The R-square for the prediction model was close to 1. But if your sample is adulterated with pork offal, we have another linear discriminant analysis model to differentiate the 3 types of pork offal with 85% accuracy. Then you can apply a corresponding partial least squares regression model to do the quantification. We were able to quantify pork kidney and pork liver with adequate accuracy, quantification accuracy, but we failed to do the accurate quantification for pork heart. The limit of detection for all types of offal adulterants was determined to be less than 10% because when we were preparing the sample, the lowest concentration of offal mixed with ground beef meat was 10%. And our mathematical model was able to differentiate all the adulterated samples from the authentic ones. So we made the conclusion that the limit of detection should be below 10%. So for the workflow, you will notice that there are a lot of mathematical models involved and some coding skills are needed. So to further simplify the analytical workflow, we designed this computer interface. So you simply click the button here to load the FTR spectrum, and all the mathematical models are stored behind. We'll do the calculation for you. We'll tell you if the sample is authentic or not. If it's not, then what's the concentration of the adulterant? So overall, the analysis can be completed within 5 minutes, which is very rapid. This is another example of how we applied Raman spectrometer, specifically a portable size Raman spectrometer for the authentication of fish or for the identification of fish species. In this study, we collected the fish fillet from grocery stores that belongs to 11 species of fish. Following a similar experimental protocol, we designed this analytical workflow. So once you have a new sample, you collect the Raman spectra of the sample. The first step will be to differentiate if it's Samanidae or non-Samanidae. This step can be done by visual analysis because morphological difference between Samanidae and non-Samanidae are pretty obvious. But if you want to make everything automatic, we have a support vector machine model being designed to do this differentiation with 100% accuracy. So if the sample is Samanidae, then we have another partial least squares discriminant analysis model to tell you which type of the 4 salmon your sample belongs to with 88% accuracy. And well, if your sample is non-Samanidae, then we have a k-nearest neighbor model that can tell you which species the sample belongs to among the 7 species of fish with 91% accuracy. So currently the gold standard method for fish species identification is by using DNA barcoding, which I will also be talking about in my later slides. So typically DNA barcoding utilize a PCR-based method. So the steps including DNA extraction, PCR, amplification, and Sanger sequencing, and then the sequence interpretation, and also matching to the library. So all these steps usually take about 2 days to complete. Well, compared to our methods, which only takes 5 minutes, so we think this Raman spectroscopy-based method have a really high potential to be applied by the industry or the testing labs. So far, the IR and Raman spectroscopy are very powerful in terms of analyzing samples in box size, which means I'm not targeting at specific chemicals and also not at trace level. Actually, both Raman and IR are not good to be used for analyzing trace level chemicals because they have really low sensitivity. But there are actually a lot of applications in analyzing food safety issues, which requires highly sensitive analytical methods. So for those purposes, we can apply this technology called SERS, Surface Enhanced Raman Spectroscopy. This is a derivative of traditional Raman spectroscopy by including additional material called SERS substrate. It's made of metallic nanostructures. So here are three examples of how the metallic nanostructure can look like. So this is a nanoparticle or nanorod. This is a dendrite nanomaterial. You can see the leaf and branches shape of the material. This is some flower-shaped nanomaterials. So on these nanomaterials, there are localized surface plasmon resonance. And once a laser is shining on this localized surface plasmon resonance, strong electromagnetic field can be generated. So once your molecule, once the chemical is approaching to this electromagnetic field, the Raman signals can be enhanced tremendously to allow this technology to detect even single molecule present in the sample. So with this super sensitive method, we can use it to analyze trace level chemicals. But for food samples, it has a very complex sample matrices. And based on the principle of SERS, everything approaching to the electromagnetic field, their signals can be enhanced. So if the sample is too complicated and your target molecule is at low concentration, those signals can be hidden. And so for using SERS to analyze food samples, we still need some effective extraction methods to remove the complex sample matrices. So the technique I used to achieve this purpose was the molecularly imprinted polymers. It's also regarded as an artificial antibody, which has a high selectivity, just like biological antibodies. The synthesis of MIPs involves these several steps. So first of all, you need to identify your analyte of interest and use it as a template for synthesizing MIPs. And then you need to identify the proper functional monomers that can form some binding, either covalent binding or non-covalent binding with your analyte of interest. After they self-assemble together, you will need to add another polymer monomer, we call it the cross-linker, to co-polymerize with a functional monomer and form a rigid shell around the template molecule. The next step is to use some extraction methods to break down the binding between the template molecule and the functional monomer, and to remove the template molecule. So eventually this material, having the cavities memorizing the specific chemical and physical properties of the template molecule, can be generated. And this is the final product of MIPs. So once the MIP is mixed with samples that containing the target molecule, they can selectively recognize and bind the target molecule and extract them out of the sample matrices. This is an example how we apply an MIP search for the detection of melamine. So as I briefly mentioned, melamine was a molecule to boost the apparent protein concentration in dairy products. The reason for people to do that was because based on the molecule, so all the black dots are carbon, the blue dots are nitrogen, white dots are hydrogen. So you can see that it has a lot of nitrogen. It contains 66% nitrogen by weight. And before the melamine scandal happened in 2008, the method the industry used to determine the protein concentration was by the gel methods, which only look at the nitrogen coming from organic compounds and convert it back to by the ratio and back to the protein concentration. I think the nitrogen concentration content in proteins usually about 14%. Well, for the melamine, it's 66%, which is way higher. So you only need a little amount of melamine to really boost up the apparent protein concentration in dairy products. So the synthesis of MIP for melamine was following the same procedures as I briefly mentioned. After the MIP synthesis, we use it as a SPE, a solid phase extraction sorbent. So we packed this into the SPE cartridge and did a typical SPE procedure to remove the sample matrices and to collect the samples that contains mainly melamine. And then we deposit this eluent onto a silver dendrite nanostructure being used as a search substrate to collect search spectra. For the data analysis, we did a partial principal component analysis to try to cluster the samples, that's prepared at different concentrations of melamine. So from the graph, we can see that the no melamine added samples was not able to be differentiated from the 0.001 millimole per liter sample, while the 0.005 millimole per liter sample was separated from these two. So we determined that the limit of detection was somewhere at this concentration, and converted to parts per million, it was about 0.63 parts per million, which is way lower compared to the maximum residue level set up by Health Canada, being 2.5 parts per million. The overall analysis was rapid compared to the traditional liquid chromatography, mass spectrometry-based methods. So our methods can be completed within 25 minutes, all the way from sample preparation to the results readout. So SIRS is a very sensitive method and good to detect trace level chemicals. But this technique is not very ideal if your analysis requires high quantitative accuracy. So for that purpose, we think maybe we can use NMR spectroscopy, because this is a technology that's known to have really high quantitative accuracy, which can do absolute quantification. So NMR spectroscopy is also a spectral fingerprinting method that can be used to analyze a complex sample matrices. And that's why it's also heavily used for metabolomics. So NMR spectroscopy collects a signal coming from the spinning of nucleus in the sample, and the spectra reflects resonance frequencies of those nucleus. Even though the NMR spectrometers now we commonly see are huge size that have to be put on the floor, development of benchtop size NMR is a really hot research area. And there are some companies that have been able to produce benchtop NMRs, and they're trying to increase their electromagnetic field to make the spectrometer more powerful. Here's an example how we applied NMR to solve food safety issues. This is actually a food fraud issue. So in this case, studies have been found that Sudan dye have been used to color the paprika powder to improve its appearance freshness. So this is made to improve or to reduce the production costs and to make extra economic benefits. In this study, we applied two types of NMR spectrometers. One is the most commonly seen one, the solution NMR, and the other is the solid state NMR. So we purchased the paprika powder from grocery stores and prepared the contaminated samples in the lab by spiking Sudan dyes into the paprika powder. And for solution NMR, we need to liquefy the sample first. So we did the liquid extraction, collected the supernatant, concentrated, and then loaded into the NMR tube, send it for NMR spectral collection. For the solid state NMR, you don't need to liquefy the sample. So we simply packed our paprika powder into the NMR rotor and added a little bit of solvent to allow the sample to be more staying in the rotor in a more homogeneous manner, and then send the rotor for spectral collection. Here's the results. So panel A are the results collected using solution NMR. Panel B are from the solid state NMR. All of these major peaks are coming from the Sudan dye. So we can see a clear dose response between the concentration and the intensity of these signals. And then we use the signals of the most intense peak to build up the linear regression curves. Again, panel A for solution NMR, panel B for solid state NMR. So both NMR spectrometer was able to provide really good linear regression model with the R-square close to 1. And we also prepared a few additional samples to use as unknown to test the accuracy of this regression models. So both NMR spectrometer had quantitative accuracy close to 100%. Limit of detection and limit of quantification for solution NMR was lower compared to solid state NMR because they have some difference in their mechanism. But I want to mention that even for this higher concentration or higher limit of detection, limit of quantification for solid state NMR, it's still useful to analyze the commercial samples because in previous studies, they've been found that Sudan dye present in commercial samples, the level can be ranging from a few parts per million, which seems to be more of a cross-contamination, to up to several thousand parts per million, which really represents some adulteration practice. The time needed for solution NMR was 35 minutes, including sample preparation and data acquisition. Well, for solid state NMR, this time was spent solely on the spectral collection. So the next technique I want to introduce is mass spectrometry. This is another spectral fingerprinting technology, and I'll say it's the most commonly used tool for metabolomics nowadays, or at least during the past 10 years. So mass spectrometry recognizes molecules based on their mass to charge ratios. So the samples being ionized as the MS source, and then there are some electric field in the instrument to guide the ionized molecules all the way through to heat on the detector for detection. So metabolomics was developed to comprehensively analyze small molecules for metabolites of complex samples. It was originally developed to study the phenotypic features of biological or medical samples, but with the reduction in the cost of the instrument, it's been extended to analyze small molecules of many other in many other fields, including in food science. So similar to SIRS, it's a very sensitive method, but it can be saturated by the signals contributed from the sample matrices. So to do a good MS data collection, most of the time, this technique is combined with the chromatography methods to kind of separate the complex sample matrices. And compared to traditional one-dimensional chromatography, there are a lot of two-dimensional chromatographies being invented for both the GC and LC. So during my previous studies, I was able to work with comprehensive two-dimensional gas chromatography. So unlike the one-dimensional gas chromatography, which only used one column, this two-dimensional GC used two columns of opposite affinity. So if in some cases, the eluent from the first column have there are several molecules co-elute together from the first column, it can be further separated in the second column. Here's a brief video showing how it works. So between the first column and the second column, there is a modulator to freeze the molecule for a few milliseconds to concentrate it before shooting it into the second column. So this really improved the molecular separation tremendously. And we use this two-dimensional GC method coupled with an MS for the authentication of orange juice. In these studies, we purchase the orange and some other citrus fruits from the market, prepare the juice in the lab, and then collect the mass spec data. After some data pre-processing, we constructed the chemometric or machine learning models to see if we can differentiate the samples based on their biological species, geographical origins, harvesting years, and also processing methods. So here's the results. Compared to one-dimensional gas chromatography, which usually detects about 100 volatiles in fruit juice, we were able to detect more than 250 volatiles using those two-dimensional methods. And the top group, the group that has contributed most to this sample matrices are hydrocarbons, followed by esters, alcohols, aldehydes, ketones, and there's some other minor groups. And then we did classification models. For these four models, we found that partial least squares discriminant analysis models worked best. It might not be showing very obviously because of the angle of the picture, but our models were able to differentiate all these different categorization criteria with more than 90% accuracy. So we further look into the loading values of the partial least squares discriminant models to see if we can find some potential markers that could be useful to further develop some targeted methods. So for biological species model, we found that ethyl butyrate and aldehydes in general are higher in orange juice versus alpha pinene, limonene, and some other terpenes are higher in mandarin and other citrus samples. For the geographical origin, we found that hexanol is higher in the sample that collected from one province in China. This might be due to the difference in the soil or in the climate of that province compared to other places, or the farming procedures. For the harvesting year, we found that hydrocarbons was higher in sample collected in 2014 compared to 2013. For processing method, we found that ethyl butyrate and ethyl octanoate aldehydes are higher in original orange juice versus beta and alpha terpineal are higher in reconstituted juice. So by original orange juice, we mean that those juices are not being concentrated and rediluted. Well, for the reconstituted juice, the juice were firstly concentrated and rediluted. So these differences can come from those additional processing procedures. So now I'd like to talk about the tools that based on DNA sequence for food integrity analysis. So the first technique is DNA barcoding. So by name, you might be thinking about the barcode you see on the products you can purchase from grocery stores. So it's kind of similar. So the barcodes on the products are unique to that product. Well, the DNA barcode to an animal or to a biological species is also unique to that biological species. So by definition, DNA barcoding is a digital system for taxonomic identification of organisms using a short standardized gene region. It's initially being used by taxonomists to describe biological diversity and build up a DNA barcoding reference library. And once the library is built up, everyone now gains access to the library to perform DNA-based identifications. This is a typical workflow for DNA barcoding. So as I mentioned, it was initially developed for taxonomic studies. So they care a lot about specimen preservation and also recording all the information. But for analysis in food science, the recording, we only need to record like where we get the samples and some other basic information. And then for the experiments part, we firstly extracted DNA, did the PCR amplification, and then you usually use the Sanger sequencing to get the sequence results. And then we compare the sequence results with the DNA barcode library to see which type of or which biological species present in our sample. Using this technology, we try to analyze the fish samples we can collect it from Metro Vancouver, because fish mislabeling or fish fraud is generally a very severe issue globally. So in this study, we collected 285 fish samples from grocery stores, restaurants, and sushi venues in Metro Vancouver region. We did the PCR barcoding, compared the results to the library, and found that the overall mislabeling rate of our samples was about 25%. So it's 1 in 4 of the sample was either intentionally fraudulent or unintentionally mislabeled. This ratio, it seems to be high, but it's actually lower compared to some other studies, even including those that's done in the eastern area of Canada. I think they found more than 30% or even 40% mislabeling. The difference between restaurants, grocery stores, and sushi bars are not that obvious, so ranging from 28% to 22%. After getting these results, we published a paper on that, and we also proposed a few suggestions for CFA, the Canadian Food Inspection Agencies, to amend their regulation and to help reduce the fish mislabeling in the market. If you're interested, you're welcome to check out that paper. So PCR is a gold standard for a lot of analysis, but there are some intrinsic disadvantages for PCR, including, first of all, it requires dedicated and expensive thermocycler that are not readily available for many food industry and also some small testing laboratories. And because based on the mechanism or based on the principle, it doubling the amplicons every cycle during the analysis. So it has a relatively low efficiency in DNA amplification. And the last one is, I think, well known if you have done a lot of PCR. PCR can be easily inhibited by some impurities, which commonly present in food samples, especially if you work with plants. So to solve some of these issues, we think maybe we can use some isothermal amplification techniques. And the two that I worked on previously was for the LAMP, loop-mediated isothermal amplification, and RPA, recombinase polymerase amplification. So both of these two isothermal amplification can amplify the DNA at a constant temperature, means that the DNA amplification happens all the time. There's no cycling required. So the amplification efficiency of isothermal amplification are generally way higher compared to PCR-based methods. For LAMP specifically, it amplifies DNA in a range of 62 to 66 degrees C. The optimal amplification temperature really depends on your primers and your targeted DNA sequence. Usually you can get the results within 0.5 to 1 hour. Well, for RPA, recombinase polymerase amplification amplifies DNA at 67 to 42 degrees C. Also, again, depending on the primer itself, you can usually get the results within 30 minutes. So using LAMP, I designed this experiment to authenticate pomegranate juice and to identify two types of common adulterants for pomegranate juice, being grape and apple. So in this study, we extracted the DNA and did the LAMP amplification. And one thing I forgot to mention is that this is really the beauty of LAMP. You can visualize the results directly by adding some dyes during the analysis. So to further simplify the analysis, or I try to remove the need of any specific laboratory instruments. So I developed this paper-based DNA extraction approach. In this method, I use a fusion 5 membrane, which contains glass. Glass is a very good material to retain DNA at certain ionic strength. I sandwich this fusion 5 membrane between 2 layers of parafilm and stack this onto a pile of absorption pad. So the absorption pad is used to absorb the fruit juice and also the washing solvents. And after washing step, this membrane is taken out and used for DNA amplification. So in this study, I designed 3 sets of primers for each type of fruit, represented by panel A, panel B, and panel C. So for each of the tube, I have fruit juice samples that containing different types of fruit juice or different combination of fruit juice. And my primers were able to pick up the right samples for all the tubes. So the overall analysis for this lamp-based analysis takes about an hour to finish, including the DNA extraction time. Compared to for traditional PCR, usually it can take up to 3 hours, including the gel electrophoresis step. And for this method, because I further simplify the DNA extraction procedure, there's no specific or complex lab instrument being needed. For the amplification, I can simply use an oven or a water bath. Here's another example of RPA. So we developed this method for the analysis of Campylobacter in chicken. So again, in this study, we use this paper-based DNA extraction method to simplify the DNA extraction. And then the amplification happened at around 37 degrees C, so you can simply hold it in your hand. Instead of using a gel electrophoresis, we tagged the primers with some tags and enable the amplicons to be detected by this lateral flow immunoassay. And this immunoassay actually provided a 10-fold higher sensitivity compared to gel electrophoresis, allowing us to detect 100 cells in the samples very easily. So this method can be completed within 30 minutes. And the amplification, as I mentioned, can happen at body temperature. It makes it very convenient to use. So for the analytical methods I've developed and introduced so far, they're generally simpler or faster to use compared to previous methods. But still, to complete the overall analysis, multiple steps are needed, and the throughput is usually not too high. So to further simplify the analytical workflow and to increase the throughput as well as reduce the cost, I started to incorporate the microfluidic lab-on-a-chip platform into the projects to make the analytical workflow better fitted for food analysis. So brief introduction about microfluidic lab-on-a-chip platform. This technique is to manipulate small volume of liquid samples in micro-sized channels or chambers. By a proper design, you can incorporate in tandem multiple assays onto one miniaturized device, making it really fast and high throughput to use. The common materials used to fabricate microfluidics are polymers or papers. Especially for paper, both materials are not expensive, are cost efficient, and especially for paper, in addition to the low cost, it's also more environmentally friendly. So by harmonizing the advantages of paper and polymer materials, I developed this microfluidic paper-based paper polymer hybrid microfluidic device for the authentication of pomegranate juice. So as I've introduced, for the pomegranate juice authentication project, I developed 3 sets of primers. And so in the design of this microfluidic device, I try to put 3 tests onto one single device. So I have 7 layers in total. This is an origami design, so you have to fold it at certain steps. My DNA extraction zone is designed on layer 4 in the center. So this is a fusion 5 material, the same material I used for the paper-based DNA extraction. After loading the sample here, after the washing steps, I will fold the first 3 layers on top and the last 3 layers beneath. So the stacking will look like this. So my DNA is extracted here. And then I will load some elution buffer from the top. So the liquid will wick through these hydrophilic areas of the paper material and wick all the way through to reach the bottom for the lamp reaction zones. So the small 3 darker dots here, darker chambers here are the positive samples, while the paler color or the whiter colors are for the negative control. That shouldn't contain any DNA samples. And this is one of the representative results. So for this, I have 3 positive samples and 3 negative samples. To further simplify the analysis and allow the analysis to be carried out in field outside the laboratory, we also assembled this portable heating pad with a temperature controller to control the temperature. So with this design, I was able to simultaneously analyze 3 species of fruits in the sample. And if you have more samples you want to analyze simultaneously, you just simply change the design of the device and it can do it for you. So the cost for each test, including the fabrication of the microfluidic device and the reagents for lamp reactions, it's about $4. So this is the end of my presentation, and here are the references for the studies I mentioned during my presentation. With that, I'd like to acknowledge my master's and PhD supervisors, Dr. Xiaonan Lu and Dr. Tao Huan as my postdoc supervisor, as well as our collaborators and my PhD committee members, Dr. Yada, Dr. Xu, Dr. Cho, Dr. Wang, and Dr. Hanner, as well as the funding agencies who funded my research and my studies, the UBC for your fellowship for my studies, and MyTax, NRC, NSERC as CPS for my research. So thank you for listening, and I'm opening the floor to any questions. Thank you.
Thank you, Dr. Hu. I think everyone in the audience will join me in expressing a deep appreciation for what was really a fascinating talk. I am personally certainly impressed with both the breadth, depth, as well as the clear commercial relevance of the work that you were doing. So thank you for that.
Thanks.
As a reminder to our audience members, I anticipate there will be a lot of questions, so you may either raise your hands and you'll be asked directly, or you may post your questions into the Q&A and I will read them out for you. I'm going to start off by reading a question that was posted by Natasha Haskey early on in your talk as our first question. So Natasha's question is, first, thank you for this fascinating presentation, Dr. Hu. I am wondering if you could comment further on adulterated olive oil. Is there work being done in this field to resolve this issue?
Yeah, that's a great question, because olive oil is also one of the most commonly adulterated products that you can find in the market. There are a lot of studies being done to solve this problem, and I will say one of the most powerful tools to differentiate adulterated olive oil from the authentic ones is infrared spectrometer. So infrared, so all the oil or the fat chemicals, molecules, they have really strong signals in the IR range. So IR can really differentiate the saturated fat from unsaturated fat, and those different fatty acids. So those are the key molecules that you need to know when you're differentiating authentic olive oil from the adulterated ones. Yeah.
Great, thank you. We have a second question here live from Shirley Yang. Shirley, go ahead, please.
Thank you. Thank you, Dr. Hu, for the interesting talk. I really enjoyed it. You have gone through lots of analytical techniques. So I wonder which technique is the best to solve the food integrity issues? If you are only allowed to acquire one instrument, which one do you think is the most useful one?
Yeah, that's a great question. Yeah, there are so many different analytical methods, and I've been using a lot of them. There are actually more techniques that I haven't been able to try by my own. But as I've mentioned, all the different analytical methods have their own strength and weakness. So it really depends on what kind of application you are trying to use the method for. You need to select the method very carefully. So for example, if you want to do very accurate quantitative analysis, and you really don't want to use surface enhanced Raman spectrometer, because that's a method that has caused a lot of headache around among the scientists to get very accurate quantification. So if the budget allows, I will really highly recommend mass spectrometer, because this method is very powerful to detect trace level chemicals and also differentiate a lot of different chemicals. But you really need to have that budget for it. So as I mentioned, each technique really has its own strengths and weakness. You have to select that wisely based on the goal of your research.
All right, I'd like to step in with a question, if I may. Early on in your presentation, you were talking about a technique for detecting mislabeling of fish species, and it looked like that was something that could actually be applied rapidly in situ, which seems to be like phenomenally relevant given the prevalence of fish mislabeling. You referred to an accuracy rate of, depending on the fish species, I think it was 88 to 91%. And I'm wondering, for practical commercial application, is that a sufficient accuracy rate, or are there standards around what would be considered a sufficient accuracy rate? And if it would be required to further increase accuracy, what might you be able to do in order to kind of push that needle a little bit further?
Yeah, that's a really good question. So actually, that's the study I used Raman spectrometer for fish species identification. That method was used in an untargeted manner. So for untargeted analysis, there's not a lot of standards being proposed or being developed to guide you through what kind of accuracy you really need to achieve. But definitely compared to the gold standard, the DNA barcoding method, the accuracy of Raman spectrometer at this stage, well, at least in my study, because I only had that limited amount of fish samples collected by myself. So it really cannot compete with DNA barcoding. But for your second question, to how to improve the accuracy, the best way is to enlarge the sample size. So to collect all kinds of fish you can get from the market and from different years, harvested in different seasons, and you really have a comprehensive library to make the samples representing all the relevant samples that you can get from the market. And so with that, you will be able to build up a more comprehensive library. And once you have a new sample, most likely this sample should be similar or somehow represented in your library. And then with this kind of library, you will be able to do more accurate identification.
Okay, so ongoing work to do. And if I could ask a follow-up question then. Yeah. So congratulations first on your appointment at Carleton. That's very exciting.
Thanks.
We've got a really neat research trajectory ahead of you, but I'm curious, and I'm sure you were asked this question when you were interviewed for Carleton.
Yeah.
What are you going to do there? What new projects, what directions are you going to take this?
Right. So I propose to do about 3 research directions I will pursue. I will be continuing to try to develop novel methods for food authentication, because food authentication is really an issue that's, I won't use the word unsolvable, but it's really difficult to solve. There are all kinds of practice to make the fraudulent products and all kinds of ways to hide those fraudulence. So we really need powerful analytical methods to be able to differentiate the new types of adulterated samples from the authentic ones. And another research area I will pursue is still the detection of trace level chemicals, because a lot of food safety related chemicals or even microbes are presenting in food at low concentrations. So we really need sensitive methods to allow the detection. And previously, majority of these methods depends on mass spectrometry, which, as I mentioned, are expensive and requires highly trained personnel to operate. So I wanted to develop some sensitive sensors that can be applied in the field or more user-friendly to allow the food industry to apply. Another research area is I would like to use metabolomics to help study the food toxicology, or how the processing procedures are changing the overall food chemical composition, and try to help improve the food production procedures.
You are going to be a busy researcher.
Yeah, I think so. But I don't want to start all the three areas at the same time. So I will gradually move in, because for the metabolomics part is more of a new research area for me. So I will gradually shift to there when I have more students, for sure.
I'm certainly going to be fascinated to watch what comes out of your lab. I see we've got a question next from Dr. Young.
Hi, thank you so much. I'm fascinated by all of the different ways you've managed to take these fairly intense chemical techniques and make them accessible so that they can be used more widely. I wanted to ask something which is a little bit less focused on your science. I too went through UBC Vancouver. I wanted to ask a little bit about sort of maybe the non-science. Science is challenging, but sort of the non-science challenges you feel that you may have faced as you move through this, and how you worked through them. Does that make sense?
Yeah, so I guess like how the gender or the culture difference, those kind of, I think so I'll talk about some of my personal experience. So I know that a lot of girls, a lot of females, if they during their growing up, when they say they want to pursue career in STEM, or even pursuing professorship, they will get a lot of rejections, because it will be a very busy career for them. But I think I was pretty lucky during, in my family, my parents and everyone in my family are quite supportive, and they don't really differentiate girls and boys when I was growing up. So I got a lot of support to do whatever I want to pursue. But I do, when I was doing bachelor study in biological science, I do see more male students compared to female students in my undergraduate programs. I think the situation in food science is a little bit better, because a lot of girls choose food science because they like to eat. That's what I heard very often. So a lot of girls will pursue food science, and nobody really reject them, because a lot of people think food science are kind of like chef. You just do what you can do, similar to what you can do at home. But it's different, especially if you are pursuing the graduate studies and pursuing a higher educational level, you will see the number of female reducing a little bit. Especially if you're entering the academia to become a professor, you will see more male professors compared to female professors. Yeah, I think I was not impacted by those, to be honest. I know that I want to pursue this, and I know that I can do these things in like adequately to be recognized by other researchers. So I know I can do this in a good way, and then I just pursue it. Mm-hmm. So I hope the other female students or other even male students that's discouraged by some of the environment, they can try to think for themselves, like if they really want to pursue it, think about if they like this field or if they can do very well in this field with their ability. So if you think you can do a good job, then just go for it. Just ignore the voices from the environment.
Great. Yeah, I think I have given that. I've done a few women in science panels, and my advice sounds very similar to yours. I usually use more swear words for impact, but it comes out to the same thing. Thanks so much.
If I can follow up on that, I'm wondering, Dr. Hu, if you had any particularly influential role models along your academic trajectory that you might like to share, as well as how you hope to inspire aspiring young female researchers in your field.
Yeah, I think one of my role model was actually not doing any research, not a scientist. So she was my teacher, English teacher, when I was in junior high. And the first day we entered the classroom, the first sentence she told us is just, if you want to do something, just try to do it at your best. Otherwise, you just don't do it. So I always use that as a standard to see how I'm doing everything. So if I want to do something, I really try to push forward and try to do my best. And I think this really helped me to go through all the difficulties and challenges during my previous education and research. And I think I will also use this. I will also tell this to my later on my students, and hope this can inspire them to be the best of themselves, either in research or just as a human being.
Great. I see that Dr. Ralt has a question next.
Thank you very much. It was a wonderful talk, very interesting. I had a quick question. I'm going to kind of go back to the fish identification studies that you were doing. So one of the questions I have is, you identify them as, you say these are species identification, but you put things like tilapia. Well, tilapia is a group of fish, which, like, you could have Oreochromis niloticus and Oreochromis mozambicus are the two most common aquacultured tilapia. And there's a whole bunch of hybrids. What level, I guess, at a taxonomic level, a person doing barcoding would say, well, you have to know to the species level. Mm-hmm. That's important. What level can you identify to at the taxonomic level with these techniques? And being a food scientist, can you comment on where is it really necessary to understand that this is not fraud? This is, if you say this is tilapia, are you just saying this is all possible tilapia? Or do we have to get to the species level to be considered not fraudulent? It's in the food sciences industry. Being not a non-food scientist, how critical is that to food scientists and the security of food?
Yeah, thanks for that wonderful question. So in that case, I'll go back to that slide. Wait a sec. It's here. So in this case, the reason, we didn't differentiate the exact species for tilapia. The reason for I to include tilapia in my study, if you go to the grocery store, or especially sushi venue, if you purchase snapper sushi, you will most likely get tilapia from it. And I'm not sure which specific type of tilapia it belongs to. I believe in our DNA barcoding project, we were able to for sure identify the specific tilapia species. But for this Raman spectrometer based methods, we only try to differentiate the larger groups. Because even in sushi venues, if the fish the fish will only be labeled as tilapia instead of the specific biological species, the Latin name. So for the application of ramen, I would say it's more like more useful in the retail level or in the restaurant level to tell you if you are really getting the species not the species or the group of fish that you are getting. Because in the restaurant, if you buy snapper, they will not tell you which type of snapper you're getting. They will call everything as snapper. So for this kind of application, that level of differentiation will be enough. But if for taxonomic study, definitely we need more higher power of the differentiation. Did I answer your question?
Yeah, I guess the thing to follow up from that is, not being in the food science industries. I'm, if I was a biologist, I'd want to know at the species level, right? Maybe, but what is the policy? Like, how does it represent from science to policy? What is the level that policy requires identification to within Canada, say the CFIA?
Yeah, that's a good question. So CFIA has a fish list. In their fish list, they have the Latin name, the common name, and some vernacular names. They have all kinds of names that's associated with the fish species or the fish types that are commonly sold in Canada or can be found in Canada. So in their list, a lot of fish, even though they have different biological names, they will have the same common name as snapper or tilapia or halibut or salmon. So a lot of the fish species are not required to be labeled with according to CFIA, they don't require you to label the Latin name on the food products. That's not a mandatory requirement. So you can use a common name which can represent several different biological species with one name, one group of name. And in that case, there's really, I guess, no need for a super high resolution differentiation to allow you to tell if the product is authentic or mislabeled. So as long as it's not snapper, it's tilapia, then it's a mislabeling or fraud.
Thank you very much. That's very wonderful. Thank you.
Dr. Young, go ahead.
That just made me think of something in that last bit. How do you differentiate between mislabeling and fraud? Is that something that you worry about?
Yeah, that's a good question. So with all this kind of analytical methods, it's very difficult to differentiate intentional fraud from unintentional mislabeling. So we can only infer if it's a specific fraudulent practice by looking at the price of the fish products. So if the real species is at a cheaper price, for example, in the sushi venue, a lot of sushi venue use tilapia to replace snappers because snappers are more expensive. So yeah, but still, even tilapias are cheaper. It's difficult to tell at which level during the supply chain did the fraud happen. Yeah, maybe the sushi venue owners, they just purchased the snapper. They think that's the right product from a froster at a higher level.
So they got defrauded as well, or
yeah.
They picked something that was mislabeled. Yeah.
Right. So to really tell if it's mislabeling or intentional adulteration, you really need to go back into the supply chain to look at different nodes. Yeah.
Okay. Thank you.
That's so interesting. It seems to me that your work would be quite interesting to a whole range of stakeholders, to policymakers, to food service providers and retailers, to consumers. But I'm wondering what you see to be your role, as well as potential mechanisms for transferring your work to those stakeholder groups.
Yeah, I think it's very relevant to my current position at Health Canada. So my current postdoc position at Health Canada, it's founded by a collaborated project with the CFIA. So we're trying to develop some untargeted metabolomics-based methods to authenticate food samples. So once this method is being established, then the laboratories within Health Canada and within CFIA, will really use this method to analyze the market samples and to see like how the authenticity situation is going on in Canada.
Okay, I don't see any more questions from our audience here. So this is a last opportunity for audience members. If you would like to pose a question, please put up your hand or pop it into the Q&A. All right, and Dr. Hu, I would like to thank you once again for participating in our Diversity in Biology seminar series and for your fascinating talk. We much appreciate that you've taken the time to prepare your talk, to spend with us today, to answer our questions. Again, I wish you best of luck as you start your career at Carleton. So from all of us, thank you. Have a wonderful remainder of your day.
Thank you. It's my pleasure. Thanks.